Bismillahirrahmanirrahim. Assalamu alaikum viewers. I hope you all will be in very good state. Welcome to my channel Cognitive Gleam. Today we are going to discuss neuron structure, and it's really very important for us that before moving toward our topic we should have a brief introduction of nervous system. We should at least know that what nervous system exactly is. So nervous system means network of nerves, as its name indicating that nerves are present everywhere. They're transmitting signals and innervating organs, and it basically receives and processes information from sensory organs and the viscera to determine the state of external environment. Now what's coming from external environment? Of course our sensory information and internal environment, visceral information. Our nervous system carries out a complex array of tasks. It allows us to sense various smells, produce speech, and remember past events. In addition, it provides signals that control body movement and regulate the operation of internal organs. Okay, and there are basic two main important divisions of our nervous system. The one is central nervous system and the other one is peripheral nervous system. Okay, now what they consist of, CNS and PNS, what they consist of. Our central nervous system consists of brain and spinal cord. Okay, and it integrates the information and makes decisions on appropriate actions, then sends instructions to certain organs such as muscles and glands, instructing them to perform appropriate tasks. Central nervous system is also the site of learning, memory, emotions, thoughts, language, and other complex functions, and it's controlling our five senses and internal organs and glands. Okay, and now our peripheral nervous system. It consists of all the nerves outside the central nervous systems. Okay, and it consists of neurons that provide communication between the central nervous system and organs throughout the body. Okay, now let's have look on further divisions of the PNS. Peripheral nervous system can be further subdivided into two divisions. Okay, one is afferent and the other one is efferent. Now neuron of the afferent division transmit sensory. Okay, sensory, as we have mentioned above, that what sensory information is, it's coming from external environment, and visceral information from the organs to the central nervous system. Information transmitted to the central nervous system includes the somatic senses. What comes in somatic senses? It's associated with skin, muscles, and joints, and special senses like our vision, hearing, equilibrium, and smell and taste, and visceral information, okay, pertaining to the internal environment such as fullness of our stomach, blood pressure, and blood pH, etc. Okay, now we will discuss about efferent division. Neurons of the efferent division transmit information from the central nervous system to the organs in the periphery called effector organs that perform function in response to commands from neurons. Effector organs are usually muscles and glands. A neuron capable of transmitting message to an effector organ or receiving information from a sensory organ is said to innervate that organ. Okay, viewers, now efferent division can be further subdivided into two main branches: the somatic or voluntary nervous system, or autonomic. Autonomic is also known as involuntary nervous system. Okay, now somatic nervous system consists of the motor neurons which regulate skeletal muscles contraction, and the autonomic nervous system consists of neurons that regulates the function of internal organs and other structures such as sweat glands and blood vessels that are not under voluntary control basically. Okay, this, you know, it always comes in our involuntary control. The autonomic nervous system can be further divided into two branches: the parasympathetic and sympathetic nervous system, which tend to have opposite effects on organs. Okay, now body also has an enteric nervous system, and in this lecture we are not going to discuss enteric nervous system. We will discuss it in our next lecture. Well, okay, now we will move towards cells of nervous system. Our nervous system complete as a whole, both divisions. Okay, both divisions contain two classes of cells: neurons and neuroglia. Neuroglia also known as glial cells. Now what are neurons? Neuron is basically the functional unit of our nervous system, and our nervous system almost contains 100 billions of neurons. Okay, and that is the smallest unit of the tissue that can carry out the function of that tissue. Neurons are excitable cells that communicate by transmitting electrical impulses. Excitable cells are defined as cells capable of producing large, rapid electrical signals called action potential. They don't divide to reproduce themselves and transmit signals. They are irregular in shape and have number of extensions, sometimes called processes, okay, or neuroids. We will discuss it later. And there are so many neurons in the nervous system to cover all the mental ground. They encode, transmit, store information about everything from your thoughts and senses to your body movement, dreams, and memories. Now, what are glial cells or neuroglia? Glial cells account for 90% of the cells. And what it means of glia, and glia is Latin word, and it means glue. They held nervous tissue together. Their main functions include providing structural integrity to the nervous system, and it also provides chemical and anatomical support that permits neurons to carry out their function. In contrast to neurons, glia don't generate, okay, and propagate nerve impulse, and you can say that they can propagate action potentials. But recent studies suggest that glial cells may also play important role in intercellular communication, and they can multiply and divide in the mature nervous system. In case of some injury or disease, new glia multiply to fill in the spaces formerly occupied by neurons, and sometimes they cause tumor. Okay, and the tumor caused by glia, they are known as gliomas, and they tend to highly malignant and they grow very rapidly. There are 6 types of nuclei present, 4 like astrocytes and the oligodendrocytes, microglia, and epidermal cells. They are present in CNS and Schwann cells and satellite cells. These 2 types of glia are present in peripheral nervous system. And in this lecture we are not going to make any further discussion on glial cells, and we will discuss it in our next lecture. But in this lecture we will have brief introduction on oligodendrocytes and Schwann cells, what they are performing, what action they are performing. Basically, they help in creating a myelin sheet around the axons of the neuron. So it helps in insulation. Okay, such insulation enables neurons to transmit action potential more efficiently and rapidly. Okay, now what the difference is that oligodendrocytes form myelin around axons in the central nervous system, while Schwann cells form myelin around axons in the peripheral nervous system. Okay, so as you can see in this diagram, this is myelin sheet, and this is axon, and this is node of Renner wire, and this myelin sheet is creating by Schwann cells, and this myelin sheet, this is creating by oligodendrocytes. And in the other diagram, in the mid there is present axon, and around axon there is present myelin sheet. Now we will discuss the structure of neuron, and there are 3 parts of neuron structure. Number 1 is cell body. It's also known as soma or perikaryon, and 2 main extensions or processes or nerve fibers or neuroids. They are coming out of cell body, and they are 2 ends of cell body, and they are known as dendrites and axon. Now dendroid is the receiving end of cell body, and axon is the transmitting end of cell body. And dendroid basically taken from a Greek word dendros, it means tree, and there are around about 1 to 20 dendroid in a neuron. And there are short knobby structures appear on the dendroid. Okay, so there may be thousands of dendritic spines on one neurons, and it increases the surface area that the dendritic tree has available for receiving signals from other neurons. So dendrites branch from the cell body and receive inputs from other neurons at specialized junction called synapses. And what is synapses, we have discussed it in our previous lecture, so you guys may concern it if you have no idea about synapses. So like other cells, neuron also contains outer plasma membrane, an inner cytoplasm in which nucleus is present and other cell organelles are present. So nucleus is the control center of the cell. It directs the activities of the organelles which are responsible for all cell functions. And cell body also contains some typical cellular organelles such as lysosomes, mitochondria, and Golgi complex, and it also contains free ribosomes and prominent clusters of a rough endoplasmic reticulum termed Nissel's body. The ribosomes are the site of protein synthesis, as you guys all know, and newly synthesized protein produced by Nissel's body are used to replace cellular components as material for growth of neurons and to regenerate damaged axons in the PNS. The cytoskeletons include both neurofibrils composed of bundles of intermediate filaments that provide the cell shape and prominent support, and microtubules which assist in moving material between the cell body and axon. So just have a look on its diagram. It will clear its image in our minds. So this is cell body. This is the basic structure of neuron, and the axon portion and the cell body are now we will just discuss about cell body right now, and we will discuss it later. So this is the cell body, and the branches coming out of the cell body, these are dendrites, which is the receiving end, and this is central nucleus and cytoplasm, and this is the outer membrane, Nissel's body, and mitochondria. And where are the dendritic spine? Here. These small projections, these small spikes, these are known as dendritic spines. So, well, what is axon? Axon basically is an other branch that is coming out of the cell body. As we have discussed earlier that two branches come out of cell body, one is dendrite and the other one is axon. So dendrite was the receiving end, so the axon is the transmitting end. Okay, it always sends information. A single axon of a neuron propagates nerve impulses toward an other neuron or a muscle fiber or a gland. Generally a neuron has only one axon, but axon can be branched. Okay, they branch sometimes, thereby sending signals to more than one destinations. The branches of an axon are called collaterals. And as with dendrites, the extent of branching varies among neurons, and it is indicative of the amount of communication with other cells. And what is the length of axon? Its length is from 1 millimeter to 1 meter long. The longest axon in the body is the one that extends from the base of spine to the big toe, and it's 1 meter long in adult. And in giraffe, same axon is 4.5 meter long. And axon contain mitochondria, microtubules, and neurofibrils. Because rough endoplasmic reticulum is not present, so protein synthesis doesn't occur in the axon. The cytoplasm of an axon, called exoplasm, is surrounded by a plasma membrane known as axolemma. Lemma means sheath or husk. So as we have discussed earlier about the axon collaterals, that the branches that coming out of the axon, so the axon and its collaterals end by dividing into many fine processes called axon terminals. Now we will discuss about axon hillock. That what is it? It is the site where axon originates from the cell body. Okay, so the part of the axon closest to the axon hillock is the initial segment. In most neurons, nerve impulses arises at the junction of the axon hillock and the initial segment, an area called the trigger zone. So where action potential is generating, that area is known as trigger zone, from which they travel along the axon to their final destination, or you can say that it's moving toward the terminal. So the axon terminal is a specialized specialized structure to release neurotransmitter on arrival of an action potential. The released neurotransmitter molecules carry signals to the postsynaptic cells, usually to the dendrite or the cell body of another neuron or to the cell of an effector organ. The neuron whose axon terminal is releasing neurotransmitter is called presynaptic cell, and the neuron who is receiving neurotransmitter is always known as postsynaptic cell. And what presynaptic cells are and what postsynaptic cells are, we have discussed them in very detail in our previous lecture. You guys may concern it. Okay. So the tips of some axon terminals swell into bulb-shaped structures called synaptic end bulbs. Other exhibit a string of swollen pumps called varicocytes. So both synaptic end bulbs and varicocytes contain many tiny membrane enclosed sacs called synaptic vesicles that store a chemical neurotransmitter. So many neurons contain 2 or even 3 types of neurotransmitters, each with different effect on postsynaptic cell. Okay, some have inhibitory effect and some have excitatory effect, and some have both effects, and we have discussed them in our previous lecture, but we will discuss neurotransmitters in more detail in our next lecture. So many neurotransmitters are synthesized and stored in axon, but not all. Okay, some are synthesized in cell body and then transported down the axon to the terminal, okay, where they release in the synaptic cleft and received by postsynaptic cells. So when neurotransmitters molecules are released from synaptic vesicles, they excite or inhibit other neurons, muscle fibers or gland cells, like they cause muscle fibers to contract. So the site of communication between two neurons or between a neuron and an effector cell is called the synapse. Okay, what is synapse? It's derived from two Greek words. Syn means together and hapten, which means to clasp or to hold. So synapse is the area where two neurons held together. So now we will discuss about slow axonal transport and fast axonal transport. Because some substances synthesized or recycled in the neuron cell body are needed in the axon or at the axon terminal. So how they will travel from cell body to the axon terminal? For this purpose, 2 types of transport system are present that carry materials from the cell body to the axon terminal and back. The slower system, which moves material about 1 to 5 mm per day, is called slow axonal transport. It conveys exoplasm in one direction only. Okay, from the cell body toward the axon terminal. It's generally associated with movement of small soluble molecules in the cytosol. And the next one is fast axonal transport. And what is it? It's capable of moving material a distance of 200 to 400 mm per day. Uses proteins that function as motors to move material in both directions, away from and toward the cell body, along the surface of microtubules. So fast axonal transport moves various organelles and materials that form the membranes of axolemma, synaptic end bulbs and synaptic vesicles. So some materials transported back to the cell body and degraded or recycled, other influence neuronal growth. So next we will discuss about myelination. As I have told you guys before that glial cells in the CNS and PNS, they create myelin sheet around the axon to insulate the neurons. So in the CNS they are known as oligodendrocytes, and in the peripheral nervous system they are known as Schwann cells. Now both types of glial cells wrap fatty substances around axon known as myelin, and the process of wrapping is known as myelination, and it allows fast conduction of the nerve impulse. Now the outer nucleated cytoplasmic layer of the Schwann cells, which encloses the myelin sheet, is the neurolemma. A neurolemma is found only around axon in the peripheral nervous system, because Schwann cells are only present in peripheral nervous system, so they are creating neurolemma. So definitely it will only present in peripheral nervous system, not in the central nervous system. When an axon is injured, what happens? So the neurolemma is regeneration by forming a regeneration tube that guides and stimulates regrowth of the axon. So gaps in the myelin sheath called node of Frenner wire, as we have seen in the diagram of axon, that is node of Frenner wire, a gap between two myelin sheets. Okay. So each Schwann cell wrap one axon segment between two nodes. Okay, and in the CNS, what happened in CNS? In CNS, an oligodendrocyte myelinates parts of several axons. Okay, as we have seen that Schwann cell will wrap one axon segment between two nodes, but oligodendrocyte myelinates parts of several axons. A neuroloma is not present. However, because an oligodendrocyte cell body and nucleus don't envelop the axon, node of Frenner-Weyl are present, but they are fewer in number. Axons in the CNS display little regrowth after injury. This is thought to be due in part due to the absence of neuroloma, maybe. So guys, that was all about the structure of the neuron. And if you have any question or any confusion, please comment me and I will clear it, inshallah. And see you in the next lecture. And if you guys like my lecture, please like it and comment and subscribe my channel for further videos. Thank you. Allah Hafiz.